Okay, welcome everybody to the second session of the ISB's virtual conference series of 2026. Again, I'll apologize for the time zone kerfuffle. We didn't realize that the American time zones were changing, and we're really happy that 46 people are in the chat right now and able to join us despite the scheduling um, challenges. Thank you so much for all being here. This is part of the conference series that we're doing virtually to enable people who haven't been able to attend in person at the conference that's happening in Cape Town in about a month. But still, we want to be a little more proactive as the ISB and as the executive committee of the ISB to have some events to bring us together. The first session of our virtual conference this year was about a month ago, and it was on the topic of AI and biocuration. And that was a well-attended event, and people were very happy to give some demos and to hear some thoughts from each other. The concept of this session of the virtual conference is a little bit different. We're going to have a couple talks and we're going to have a panel. And the theme of today is curation beyond biocuration. And we're very lucky to have representatives from other topics outside of the life sciences. We have linguistics, we have archaeology, we have physics and natural sciences. We have mathematics and we have machine learning represented by our talks and our panelists. Let me give you a little bit of a flavor on who's going to be with us. So we have a couple lightning talks, we have an invited talk, and then we have a panel discussion. Because of the scheduling changes, I think we're going to start with some of the lightning talks. And like I mentioned before, we might do the panel discussion before we get to the invited talk to fit in with the schedules of our panelists. So I'm going to hand it over to Federica Quaglia, who is the co-organizer with me. Oh, by the way, I'm Charlie Hoyt. Hello, member of the ISB Executive Committee. I'm going to hand it over to Federica, and she can introduce our first lightning talks.
Thank you so much,
Please
Charlie.
go ahead and use the chat if you've got any questions. I'm going to stop screen sharing now.
Thank you. Hi, everybody. So we have three speakers today for our lightning talks, and they are spanning different areas of curation, not really biocuration, so archaeology, linguistics also. And we are going to start with Francesca Masini now. Francesca is from University of Bologna, and she's going to talk about her efforts in a unit dedicated to digital processing of language data and the creation of resources. She currently serves as a rector's delegate for open science research data and research assessment. And she's also co-founder of Linguisticamente, which is the first Italian website dedicated to popularizing language sciences. So thank you, Francesca. You can share your screen.
Thank you so much. Good evening, at least from here. Evening, everybody. Okay, let's share the slides. Can you see them?
We do.
Like that?
Thank you.
They are big enough?
Mm-hmm.
Great. Okay. In this lightning talk, I will very briefly introduce you to data curation in linguistics. So, what is data for linguists? So, our data is language as produced by humans. So, it can be spoken, it can be written, it can occur naturally in the world, it can be elicited in an experimental setting. And of course, there is data and data. So, we have primary data, which is the original linguistic observation, let's say, minimally processed, and then secondary data, which is a representation or interpretation of these primary data through annotations, for instance. And what is interesting is about language is that it is very, very complex adaptive system. So a single utterance, even a single word, has many layers of information like a sound, syntax, morphology, meaning, and none of these layers is the data. They just coexist, and it is the linguist that chooses what aspect to put in focus each time. Of course, there are a lot of challenges in linguistics, in data curation. So one aspect is that we need both the generalization and variation. So every concrete linguistic act is unrepeatable and unique. So we often have to generalize over that to avoid extreme fragmentation. But at the same time, sometimes differences between speakers and contexts can be exactly what we want to study. So we have to strike a balance between these two aspects. Then linguistic data is rarely self-describing. It is often enriched with annotations, but every annotation, every label, tag, or category that you use reflects some theoretical assumptions. So this means that the same data can be annotated differently. Of course, there are some standards, but believe me, we have to resort very often to ad hoc annotation schemes. And another challenge is ethics. Ethics is structural issue for us because we have to do with humans, so with individuals, with communities. So questions of consent, ownership, reuse are just not add-ons, but they are really shape from the very beginning what we can curate, share, and preserve. Finally, how do linguists curate data, or much they do so? Data management can vary across data types. So as for primary data, they obviously need to be preserved and documented, possibly in trusted repositories. As for secondary data, we really need to be very transparent and explicit about every step, every decision in their creation to ensure reusability. Reusability is a little bit of a, let's say, tricky issue because overall, as of today, data sharing and replication studies are not still so widespread in linguistics, but things are slowly improving. And finally, some insights from the language sciences, which I hope can be of any help or relate to what you're doing. So for us, small is valuable. So big data helps, but high quality small data set remain essential, especially in some fields or subfields. LLMs can be of great help. They support annotation and analysis for linguists, but they require expert human oversights always for every single data. And finally, there is this new paradigm of perspectivism. So statistical patterns matter and help us a lot, but at the same time, individual variation, even the outliers, are worthy and informative. So when your data talks back, you listen carefully. Thank you so much.
Many thanks, Francesca. We have time for a couple of questions, I would say, before we move on with the next lightning speaker. You can type in the chat if you want. Of course, if no question, we can also have time for this right after. But I see Charlie. Thank you.
Hi, thanks, Francesca. How do linguists think about making data standards so people who want to curate similar kinds of data, but in different places with different groups of people, what do data standards look like for linguistics?
Yeah, that's a tricky issue because there are some standards, even some very widespread standards. Like one example is the universal dependencies, for instance. There's a paradigm for annotation of linguistic data syntactically wise. And it is used for many different languages, so that's why it has universal in the title. But then it really depends on the layer of information that you want to analyze. So for some layers of information, there are standards like syntactic or morphological standards are pretty much used. But then when it comes to semantics, for instance, meaning, then things explode, and it's very difficult to find a standard in that way. So it depends on which aspect you're handling.
Many thanks, Charlie and Francesca. I do not see any other question for Francesca, but if we have more, I think we have a couple of minutes also after the talks. If so, I think we can move on with the next speaker. Next speaker is Vanessa Baratella. She's a protohistoric archaeologist and currently a researcher at the University of Ljubljana. with an SCA project, and her primary research interest is the study of political, economic, and social dynamics that led to the formation of the first urban entities during the Iron Age in Europe, particularly in northwestern Italy and Slovenia. So thank you, Vanessa.
Thank you. I can share my screen.
You can, yes, please.
Okay, you can see my slide.
Yes. Okay,
And
perfect.
my
Thank you.
presentation.
Mhm.
Okay, so thank you, Federica, and thank you, Charlie, for inviting me to the conference. And I know my field of research is still far from having data systematization protocols comparable to other disciplines. So I hope this is a good moment to start to adopting this good practice. So when we think about the challenges of archaeology, we often imagine excavation, dating, and interpretation. But one of the limits I encounter most often is another. And in archaeology, we don't always call things by the same name. We use different languages, and also when we use English for publications and conferences, we don't use a standard vocabulary. In archaeology, research, fieldwork, and publications cross national borders. And this is especially true for the Iron Age, a period of strong connections across all Europe between 9th century BC and 4th century BC. Objects, technologies, and cultural practices circulated widely between different communities, and it was a real global system. But in many ways, the past we study was highly interconnected, but however, the way we describe the past is often fragmented. Archaeological knowledge is produced in many different languages, and each scholarly tradition has developed its own terminology to describe our data, so artifacts, structures, and cultural phenomena. As a result, the same concept may have different names depending on the country or the research tradition, sometimes even within the same language. For archaeologists, this can already create confusion, but could be accepted because it's a linguistic limitation. But for digital data, the problem becomes very significant. Today, archaeology tries to increase digital database, share repositories, and also international research infrastructure. But we are at the very beginning. If terminology is inconsistent, data cannot easily communicate with each other. This is why organization tools such as control vocabularies, thesauri, artifact database, and ontologies are becoming increasingly important in archaeology, and we have to work on it. The goal is not to eliminate linguistic diversity, because it's also impossible, but diversity is one of the riches of archaeology. But the real challenge is to build bridges between languages and terminology to systematization of the data. If Iron Age society were connected through networks of exchange, maybe also our archaeological data should be connected as well. Thank you.
Many thanks, Vanessa. We have time for a couple of questions for Vanessa as well. So you can type in the chat if you want to ask her something more about her work. Okay, I see a question from Charlie in the chat.
No, someone else ask questions, please.
But I see. Is there a typical style for controlled vocabularies or thesauri and ontologies in archaeology? Like, do people love owl, for example?
Yes, the problem is not so shared in the archaeological community, because it depends also on the chronological framework that we are talking about. because this kind of vocabularies are it's okay for Roman period or medieval period. It's quite different for Iron Age because we have different typologies of materials and different problematics. And probably we have to try to speak more and try to obtain something that could be fixed okay for everyone. And for now, we have this limitation.
Thank you, Vanessa. I see another question in the chat for you. Are there current approaches for applying AI methods to normalizing artifact descriptions across languages?
Yes, we have to try to do this, and we try also to apply for a call, and we proposed a project also with some bioscientists, so that could help us in this way because we are so not expert. And so we hope that we can use AI methods and also digital archives to fix that problem. So also for normalizing the description could be very useful.
Many thanks again, Vanessa.
Thank you.
So now we are going to have our last speaker for lightning talks is Sebastian Duesing, a PhD student at State University of New York at Buffalo and an ontology consultant at La Jolla Institute for Immunology. And he's also interested in open source tooling for ontology development. Thank you, Sebastian.
Thank you, Charlie and Federica, for having me here today. I'll go ahead and share my slides. There we go. All right. Yep, my name is Sebastian Duesing. I'm affiliated with the La Jolla Institute for Immunology and SUNY University at Buffalo. And I'm here today to talk about curation on the biomedical side with the core ontology for biology and biomedicine. In my work at La Jolla Institute for Immunology, I and my colleagues work on the Immune Epitope Database, which is a freely available resource that catalogs experimentally derived immune epitopes for diseases, allergens, and transplants. The IEDB has a team of PhD-level curators that have curated many thousands of publications into a set of highly structured fields. A lot more than you can see in the screenshot on the right here. It just gets really long and hard to fit all in one slide. The IDB fields that we collect are mostly not free text. The field values are mapped to ontology terms from a whole bunch of different ontologies, like OBI, the ontology for biomedical investigations, to talk about assays and the devices that those assays involve. Uberon for anatomy parts like organs. The cell and cell line ontologies to talk about the specific types of cells that are being assayed, and more. Ontologizing the field values in the IEDB enables hierarchical queries and improves the quality control in curation, and allows us to interlink IEDB data into the wider data ecosystem in the biomedical data sciences. Historically, we faced the problem that not all of the ontologies that we rely on were particularly interoperable. Linking to terms for spleen and splenocyte is great, but we'd ideally also want there to be a relation between those two terms. And in the past, that hasn't always been the case, because those terms for cells and the tissues that they're a part of weren't always developed by the same people, weren't always developed by people working together, and so were axiomatized in different ways. So we didn't always get the reasoning power out of ontologization that we would have hoped for. However, over the last couple of years, there has been a big push within the sphere of biomedical ontologies that we work within, the OBO Foundry, to adopt the core ontology for biology and biomedicine. The OBO Foundry is a diverse set of ontologies in the biology and biomedicine domains. A lot of the ontologies, maybe pretty close to all of the ontologies that we use in the IEDB are OBO ontologies. COB is a single high-level hierarchy that contains the highest level terms of all OBO ontologies and ensures that they're interoperable and logically consistent. The adoption of COB across the oboe foundry is very much a work in progress, but it will make life a lot easier for resources like the IEDB that rely on lots of different oboe ontologies to map the data they curate. All right.
Many
I'm happy to take
thanks.
questions here or in the chat or over email. Thanks all of you very much for your time.
Thank you, Sebastian. Of course, we have time for questions for you as well now. You are free to type in the chat. Harry, you can talk.
Oh, sorry, that was accidental, but
Oh,
I love the
okay.
resource, so
Great. So glad you like it. That's awesome.
I see another raised hand, Ginny.
Thanks, Sebastian. This is really cool to hear what you're working on. So I come from an immunology research background and am using bioontologies in sort of an applied way. And I had about 5 minutes last week to look for a term for antibody and was really surprised that I would think coming from that area of research that that would be a term that would fit in very nicely in an immune ontology, but I couldn't find one. So I was just wondering if that's one of the challenges you're tackling in your work, because it would seem to fit.
It is. One of the common problems in open source ontology is that the coverage of domains varies widely based on all sorts of weird non-scientific factors that are, anywhere from personal to technical to funding related.
Hmm.
And so it so happens to be the case that there is no central singular immunology ontology. And and in some ways that's like kind of really awful for people who are looking for a single centralized resource for stuff like antibodies. So these terms are kind of scattered to the four winds. That's one of the goals of implementing COB is that if we can align all of the different ontologies to which interdisciplinary fields terms are scattered, it will be easier for people in those fields to find and use those terms. But again, very much a work in progress. And right now, terms for antibodies are scattered across the gene ontology, the protein ontology, and a whole bunch of other places. So it's very much not a resolved problem yet, but
Well,
hopefully in a few years there'll be more coordination on that front.
yeah. Okay. All right. Thank you.
Thank you.
And I do see that we have another question for you in the chat, Sebastian. Have you tried to use LLMs to map the free text metadata to ontologies trained on these curated data? If so, how do they perform?
Yes. So there's been a lot of work going back to, I think, 2007 to use any variety of machine learning tools to curate IEDB data. For a long time, that was kind of a no-go. We found that because we have a very, very low tolerance for error, that we weren't getting significant results for a long time. But, the massive improvement in machine learning and LLM technology over the past few years has really been a game changer. We are currently working with a major tech company who's working pro bono with LJI on LLM implementation in IEDB curation. Um, and we found over the past few years that the technology is much better suited than it has been for a long time. So there's progress in that area.
Many thanks again. Um, well, so I do not see other questions in the chat. But if so, many thanks again, Francesca, Vanessa, and Sebastian, for such great lightning talks. They provided a broader perspective on curation efforts beyond the biocuration, and it's really relevant and interesting. I guess that now we can move on with the panel discussion, and Charlie is going to introduce our panelists today.
Yep, thank you, Federica. So we have four panelists, potentially five. One of them has not been able to join us yet. So I want to make a quick introduction. We're going to do a round robin with a primer question to get everybody's ideas flowing, and then we'll have kind of some open discussion. So as this is happening, please feel free to write questions in the chat, and I will try and moderate, and let's hear from our panelists. So our first panelist is Damiano Testa. He is a mathematician at the University of Warwick, and I'm going to keep the introductions a little bit shorter because I think the first question we're going to ask everybody is how they came into the curation adjacent world in their field. The second person is Gavin Farrell, who's at University of Padua, and he's going to be representing machine learning as a discipline, though Gavin has worn many hats in his career and has a lot of perspective on other things as well. We also have Sami Permin-Kenza, who's at University of Southampton and with the PSDI, the Physical Sciences Data Infrastructure in UK. And so she's going to be talking about the natural and physical sciences. And as an extra stand-in, we have, hold on. We have Sushma. I'm sorry, Sushma. I'm going to do my best to pronounce your last name. It's Natiani, Sushma Natiani, who is working in agriculture and on sort of biology adjacent things. So I want to go in a round robin in the order that I've introduced you and ask, how did you get into your fields? And then how did you become a curator or a person who works as a data steward or something like that? And is there a typical path towards becoming a curator in your field? Is it something that people stumble on, or how did you get there? So, Damiano, please start us off.
Yeah. So, hi everyone. Can you hear me? Hopefully, yes. Yeah. Okay.
Yep.
Great. Yeah. So I'm a mathematician. I've been a mathematician for most of my life, and then during the pandemics, I've become interested in something that is called formalization of mathematics, which means using a computer to verify mathematical reasoning. And that is the connection with curation. So the curating proofs of mathematical statements is my link to curation. I'm one of the maintainers of one of the biggest libraries that contains proofs of mathematical facts. And even though this kind of things existed for a really long time, it hasn't made its way into math departments until very recently. So I wouldn't say there is a common path to becoming a curator in this sense. But a lot of people just start playing around with these tools that allow you to verify your reasoning. They get they like what they do, and then they decide to continue doing it. There's no real recognition for them yet, but there's been a huge amount of interest from AI companies recently that has brought a lot of funding, and so potentially also a lot of ways into this path. But currently, there is no real sort of official way of getting into it. I think that was the question.
Yeah, perfect. Thank you, Damiano. Let's move on to Gavin.
Hi, thanks, Charlie. So I'm Gavin Farrell, based at the University of Padua, doing a PhD in machine learning, best practices in the life sciences, which is where I'm bringing the machine learning hat in. So originally I was a biochemist, wet lab trained, transitioned into biomedical genomics, and then I worked for a couple of years at Elixir Europe, which is a life science data research infrastructure. And through that, actually, I got exposed to biocuration for the first time with some people in this group here. So Federica Quaglia, who I currently work with at Padova, and also Jen Harrow, who was an old colleague of mine at Elixir Europe, where there was this biocuration focus group. Out of nowhere, I kind of got pulled in and sucked in, and I got exposed to the really cool world of biocuration. Got to attend some conferences, International Society Biocuration ones. And it was a really cool field. As I progressed through my time at Elixir Europe, it was a lot of community management, a lot of wearing different hats, but it wasn't as quite hands-on the science side. And I decided to go back and for more punishment and start a PhD. And with that, I ended up doing machine learning best practices in the life sciences. So again, transitioning into a different field. It's quite an emergent field for kind of curating artificial intelligence and machine learning methods. It is still life sciences adjacent and very close with my focus. But there's a lot of cool stuff happening and a lot of international working groups, but established career pathways are still not exactly there. There's a lot of traditional research data management and research software steward roles, which are kind of coming into this space. But for explicit roles in this area, because it's quite interdisciplinary, not quite established yet.
Thank you, Gavin. Next to Sami.
Hi, everyone. So mine's been a bit of a wiggly path. So I actually did my undergraduate in computer science, and then I worked as a developer for a year and decided that wasn't the corporate world wasn't really for me, and I liked developing, but only really for research purposes. So I'm about to do my PhD, and I did a PhD in web science, which looks at it's like a blend of sociology and computer science, and it looks at the socio-technical aspects of things like the web, where actually the human intervention is just as, if not more important, in how the software is progressed. And I guess all the common area across the three projects I work in is actually the human element. And I'm always extolling the virtues of the human requirements for data curation, data management, data stewardship. This isn't something we can purely do with machines. But I ran the AI for Scientific Discovery Network for 4 years. And again, data quality, lack of access to data was something that just came up constantly. And during my PhD, I also looked heavily at electronic lab notebooks and data capture and data processing in the physical sciences. So I guess a lot of my initial work was sort of the pre-curation, sort of around more the data collection side of things. And then I moved into the physical sciences data infrastructure. So we have created a big infrastructure for the physical sciences in the UK, which has included creating and providing access to highly curated data sources, trusted data sources for people to use. And again, kind of a lot of work around fair data, AI-ready data. But also I run a project called CASDAR, the Careers and Skills for Data-Driven Research Network, which is domain agnostic, but it is looking at building a network of data stewards and the importance of actually having data stewards, and that, again, that human aspect in actually stewarding our data through the research projects, and the human effort in making sure our data is well managed all the way through the research data lifecycle. So all those things are really, really important. I also am really interested in the semantic side of things. So again, I've done work both in the physical sciences, but also actually agriculture in linking up relevant ontologies with data sets and trying to actually create the data sets to make sure that they actually make sense and are machine readable and have appropriate metadata. So yeah, quite a lot of different spheres, but all around making sure that the data is better quality. There is no real obvious pathway into what I do. I've kind of fallen into it. Data stewards, there aren't actually that many of them in the UK. They're becoming a bit more prevalent in certain areas, but particularly in the specific domains, you often end up with people like me who get very into the idea of, research data and data quality and data curation, and sort of end up falling into those adjacent paths.
Great, thank you, Sammy. And our last panelist, Sushma. You're on mute, though.
So I started my life as a microbiologist, studying microbial genetics, and then moved into molecular genetics, did a lot of gene sequencing, gene expression analysis, and then moved to plant genomics. And then we moved to plant genome sequencing and transcriptome sequencing. So that work we still continue. So last year we sequenced the chia genome and chia transcriptome. Before that, we participated with strawberry and eucalyptus. And since 2012, I have been part of the Grameen database, that is a comparative genomics portal for plants. We host over 100 plant genome browsers, and I'm the lead curator for the pathway portal of Grameen, so that is the plant reactome. And so I lead and curate and supervise the curation of the plant pathways. So for me, I had a long history of doing plant experiments, microbial experiments, and I continue to do still the plant genomics research and do curation on site.
Many thanks, everybody. So I have another question for you now. What resources do you work on, and how do you do curation? Also, how do you think that curation will do to advance in the next decade? Guess we can start again from Damiano.
Okay, yeah. So, so right now it's a lot of manual and human supervised endeavor to curating mathematical proofs. So, artificial intelligence is really not able to sort of give quality outputs yet. It certainly improved a lot in recent years. In the past, maybe 2 or 3 years ago, I would have said it's never going to work. Now I'm going to say it doesn't work right now. So hopefully in the future, AIs will improve and it will be possible to use directly AI input. But right now, even very simple tasks are sort of out of reach of current cutting edge AI models. But as I said, this is a very rapidly evolving field, so very likely they will get up to speed. And then there's, so this is sort of local things. Mathematical data also has to fit globally, so you need to be able to interact with different definitions at various levels from various different angles. And so there's also the need of a sort of a holistic view of everything that is going on. And I think that'll take even more time for AIs to reach, but hopefully it'll be there. But right now it's mostly human experts that look at things and try to sort of think in their heads how it would fit with other parts of what they know. And then when enough people are happy with that, then it gets absorbed in the sort of core language, and you move on to doing other things.
Gavin.
Hello. Yes, so for the service that I work on, largely I'm working with the DOME recommendation standard, which is a disclosure framework for artificial intelligence machine learning methods. And that covers around 20 questions, and it's a structured schema then covering 4 different areas of the method. Currently, it's largely manually curated for all of those entries. People, if they have a publication, they can come to our service and they can manually annotate those in support of their publication methods disclosure in a structured manner. But we also do direct curation of those ourselves. We found it's quite labor intensive. So over time, we've actually been extending our work to explore scalable curation approaches. So currently we're looking at a triage model to identify machine learning methods and relevant publications going backwards in time. So from BiodiversityPMC as the literature source. And we're also working with the AI for EOSC project, where we are employing a open source large language model to actually use the literature source once identified and actually scale that curation in our standard format, which has been quite successful. Going forward, looking to the future, where I see things going, well, definitely scalable curation with our artificial intelligence methods. I know a couple of people have already mentioned are becoming more and more reliable. And as things go on, I see this definitely for our field, which when you're trying to source this manual effort and people don't see the payoff to actually manually annotate them themselves, or they may not have the expertise, the LLMs can be incredibly powerful for this. So that's largely where we see our field going, and helping translate those machine learning methods and publications into structured, just reproducible and reusable methods.
Thanks, Gavin. Samantha.
I've started to put a few things in the chat because PSDI is quite a big project and there's a lot of different things. It's so hard. I'll try and cover them fairly efficiently. So as part of PSDI, as I said, we provide access to a lot of trusted data sources. So some of them have directly come in from the Physical Sciences Data Science Service, which we have folded into PSDI, and then we have a fancy cross data search that runs above it so that you can actually now use that to look at the things you want to find across all of our data sets. And we also link in with the Optimade API. So I have put a link in the chat that just sort of has all of those different resources there because there's too many to really cover. I would say kind of a big shout out to the physical chemistry properties data collection, because this is one of my amazing staff members, Matthew Partridge, actually created and curated from scratch using a bunch of different data sets and really fleshed out to try and make it as robust and complete as possible. And they're now working on some further versions of that. So that's kind of where we work around the data sources. We're also working on tools around sort of, I guess, again, that maybe sort of pre-curation stage, but we're working on a lot of tools around reproducibility in organic chemistry. One of ours is called the Glorious Plot device. It's built on the work of Frank Glorious. So it's to map your different reaction conditions and see the different results you'd get from the different reaction conditions. And then you can actually export that out as an RO crate. So again, trying to actually provide people with a way to get data out in a much more machine readable, reusable fashion. We've done quite a lot of work around RO crate as well, looking at it linked in with Electron Lab notebooks and using it as the ELN file format to transport data between them. We also work quite a lot on Electron Lab notebooks, which for anyone who joined earlier in the meeting, me and Charlie were talking about. So again, we're looking at how we can leverage AI tools to actually get people to create and sort of capture more data at the beginning in a better form. Because again, a lot of the big issues is, everyone talks about fair data and getting their data at the end and sharing it. But actually, we find that a lot of the problems start at that kind of creation stage. And actually, there's so much you can curate incomplete, inconsistent data that wasn't created or captured well in the first place. So we're trying to mitigate a lot of that work kind of at the beginning. I do see a lot of integration with AI methods coming our way, which I guess with the caveat that I definitely don't see them replacing humans. I still think the human aspect of data curation is really, really, really important, and the domain knowledge that humans bring. But I think we need to work out how to leverage the best use of tools with people so that we can actually streamline some of these processes and then bring in the human experts where we need to. We're also we're collaborating with the NFDI around this initiative called Ontologies for Chem, which Charlie was also part of our meeting last year. And again, we're looking at kind of a canon of the chemistry ontologies. So what are the chemistry ontologies that everyone's actually using? Can we then make sure that they're well curated and up to date and everyone can find them? Because again, you've kind of got this problem that everybody uses slightly different things. We're not going to persuade people to all use the same thing. And actually, to an extent, depending on their needs, that's not going to happen anyway. So how can we kind of have these places that bring the relevant information together to at least make it robust and all in one place for the community?
Thank you, Samantha. Now it's Sushma's turn.
So we do a couple of different type of curation. So we try to improve gene curation. So we try to improve the gene models. And so anytime we find a supporting different evidence from transcriptome or proteome, and there is a possibility of refining gene model, that is one part we do. The second part we do is improving gene annotation. So we look for the evidence from literature and try to link the literature experiments and improve gene annotation. A lot of the problem in our field is that more than 50% genes in most crop species, they are still in the unknown category, and there is a lot of opportunity for improving gene annotation. More recently, we have also used transcriptome, proteome, metabolome, and other type of omics data to improve gene annotation. So once we have a good gene annotation, then we look for the second layer, that is gene-gene interaction, and how to map those genes and associate those genes with biological processes and pathways. And so for the pathway curation, we share the platform and tools that were developed by Human Reactome Project, and we use Reactome Curator tool to curate plant pathways. We still share the central database with the Human Reactome folks, and so that we don't have to create things that are already there. We use a lot of ontology. So gene ontology, for example, we use a lot. In addition, we use plant ontology, so plant developmental ontology, trait ontology, that are developed by the Plantium project. So we try to have our data to be more fair and interoperable. It is available to community for free of cost. So we adhere to the fair and free data availability practice for plant reactome groups. Our data is integrated with UniProt. We are integrating our data with the Gramine and Plant Ensemble. We also represent some of the tools with the NASA visualization app, so where they can access plant pathways and also access the analytical tools that are available through our platform for doing the omics data analysis. So we try to be collaborative, and we are one entity in the bigger ecosystem of public databases. We connect when possible to other information that is already available in other databases. For example, for the detailed information on proteins, we connect to UniProt. For genes, we connect to Gramine and Ensemble portal. For chemicals, biochemicals, we connect to CABI and PubChem. And we do the detailed phenotype and trait and plant pathway curation in plant reactome. We bring the EBI gene expression atlas that can be seen within the pathways from the EBI. gene expression atlas, and similarly, they also show our data there. So we really work in the bigger ecosystem of the genomics, breeding, and pathway databases.
Many thanks also to you, Sushma. I might have another question for you. What are the current challenges and next steps for your field?
So I think it's still, I'll say that the two challenges, I think one is the accurate improvisation of the gene annotation. That is still the biggest for the crops. And then I think the second one is that we have many heterogeneous type of data. So we have a lot of genotype data and high throughput phenotype data and trait data, a lot of data coming from the environmental aspect, how plants respond in different environment and under different stresses and different association with the microbiome and other life forms. And so integrating that data and developing a more complex conceptual frameworks is a big challenge, especially connecting like all these data and making sense. That I see is a bigger challenge. And we have formed a consortium, is a Egg Biodata Consortium. So I serve in the SC committee of Egg Biodata as well. We are consortium of 40 genomic breeding and pathway databases, and we work on the FAIR policies. We also look into the common tools, including use of the LLM and AI in the biocuration, and what we can learn, what we can share together, improve, and evaluate some of those tools.
Thank you, Sushma. I will ask now Samantha, since I see that you have to leave soon.
Oh, sorry. Could you repeat the question as I was typing out my long message in the chat?
No worries. What are the current challenges and the next steps in your field?
I think the current challenges, firstly, is just how much data we're producing and the capacity to sort of keep up with that. I think we're very keen to push again towards machine readable data and data that is marked up using ontologies and leverages the power of semantics. But the actual domain expertise to identify the relevant ontologies and the relevant terms to actually do that is really, really tricky, because again, there are so many different ontologies in the physical sciences sphere. And actually, even just finding the relevant ontologies to categorize the physical sciences and describe the physical sciences was a challenge in itself, let alone kind of moving over to that. And then the fact that everyone does things differently, and it's quite hard to actually get people to do things in the same way. But I mean, part of PSDI is very much trying to bring together the best uses of tools, resources, and people. So we're always trying to look at almost what are the wins, where can we reuse existing tools, existing data, where can we bring together existing services. For example, we've created a data conversion service that helps people convert between different types of scientific data, because interoperability is a massive, massive problem in the physical sciences. But again, you can't always persuade people to use the same data formats. And again, depending on the use cases in their work, nor should they. But then you do end up with that issue that people often then can't get the data in the format they want to, collaborate with other people, use different pieces of software. So again, we're trying to identify those sort of big challenge areas and then create the tools and services, or bring together existing tools. So in this case, we brought together a load of existing conversion tools and layered a load of functionality on top of it to try and make things easier for people. And again, we're always so I did put the PSDI website in the chat. We're always very keen to hear any sort of feedback on those resources. And if there's things you think that they're missing or they should do, please do get in touch with me and tell me, because ultimately we're building them for this community.
Many, many thanks, Samantha, also for joining.
So I'm going to have to head off now, but it was lovely to meet you all. I did put my email in the chat, so if anyone wants to follow up with anything, just give me a shout.
Thank you so much again. Bye.
Bye.
I'm going to ask Damiano now.
Yeah, so I guess going from sort of immediate to further away, I think one of the biggest challenges right now is that there's a lot of contributors to our sort of repository of formalized mathematics. And it takes a lot of time and reviewer effort to make these contributions sort of feasible for being admitted into the database. And there aren't just enough people that are willing or able to do that. So that's, I think, one challenge. In fact, this has been made worse in recent months by the advent of LLMs and artificial intelligence, because people are producing a lot of very low quality proposals that sort of require even more effort from the maintainers. So that's a negative of artificial intelligence and LLMs on my side. On the other hand, there is the hope that it will improve and will therefore make everyone's life easier. And in particular, it would be great if it were able to bridge the gap between what mathematicians have done since forever of their ways or our ways of communicating mathematics to other humans, which is very implicit, very often imprecise, even though it conveys the right ideas, and being able to convert that into something that is actually correct and really is the meaning of what people are trying to communicate. So if LLMs and artificial intelligence can sort of make the conversion between the informal text that mathematicians produce and convert them into actually machine verifiable reasoning, that would be a huge advancement for the kind of things that I'm doing.
Thank you so much, Damiano. And last is Gavin.
Yeah, so when curating artificial intelligence and machine learning methods, they're quite reliant on the underlying assets, which are the AI datasets and models. And one of the biggest things in this field is the consensus and usage of kind of metadata standards. So for example, for your models, you might have things like the Fair-Fermel schema or AI datasets like the Croissant standard, but these are not widely used and they're quite emergent and evolving over time. So there's a lot of these assets that aren't marked up in any kind of standardized schema formats or metadata. Which is quite a challenge. And additionally, even when they are, the use of fixed vocab and ontologies for even filling those fields is quite weak for this kind of work, making it even harder to curate. And even when they are kind of filled and these assets are kind of marked up and annotated, and you can actually mark up these methods in, for example, our DOM registry for the methods disclosure in a standardized way. The resource diversity of where people are actually storing these is quite immense, and there's no kind of really good place yet for life science machine learning methods where you would deposit your AI datasets or AI models. There's stuff coming out of the European Bioinformatics Institute quite recently. They've put a new skin on biostudies to actually host models and datasets. But the underlying metadata schemas for those is a challenge as well and aren't quite fixed. It's very loose, which is a good start, but community consensus is really needed in this space that will really kind of help and accelerate the ability to curate and standardize all of these methods to make the methods more accessible, reusable, and reproducible. And of course, then there's the diversity of checklists. These are proliferating, and really, again, community consensus needs to come into place to start standardizing and creating crosswalks across these to make methods more valuable, both within domains and across domains. And maybe things like the European Open Science Cloud bringing together different communities where they are, again, possibly moving in different directions and more convergence could come together through those efforts.
All right, thanks, Gavin, and thanks, panelists, for the beginning of our round robin session. So I mentioned in the chat the next part of we have about 15 minutes, almost 20 minutes left. Please feel free to ask questions in the chat or raise your hand, and I will relay those to our panelists. The first one was from Sue Bella, and she wrote in the chat, are there ways that we can support curation efforts across our fields? And are there things that we can do as a community to help each other learn from our different experiences and leverage from our experiences? And I think when she says our fields, she means across the different fields that are represented here.
Yeah, Charlie. I meant, because I'm listening to everybody talk and I'm like, yeah, that sounds really familiar. Standards aren't standard, resources are scattered. Yeah, you're developing ontologies, but if the people developing them aren't coordinating, then you end up with 10 overlapping, incompatible ontologies, and the term you want isn't actually in any of them. So yeah, just sort of if people have thoughts about ways that we can try to help each other learn and not keep falling into the same giant ditches over and over again. I didn't mean to kill the conversation.
Can I redirect that question a little bit towards Damiano? So lean is like, this formalism that you mentioned, and you're building a database of all of these proofs that have been formalized there. What are the things that are similar about the way that you do that to what a lot of the other speakers and panelists and people in the chat have talked about? They're talking about ontologies. Is there something like that in your field that is a place where you might want to standardize the same way that the biocurators and the physical science people are standardizing?
So I was actually wondering about this because I don't really know, or I don't think I really know what an ontology is. So what we end up doing is basically mathematicians tend to write very long, complex proofs, and in order to sort of verify them, you need to break them into really small, tiny pieces, and then the computer checks that those tiny pieces fit together in the right way. So the repository that we're building has a lot of these very small things, and then a lot of things that are built out of these very small things, and then a lot of things that are built out of these bigger things, and so on. So there's all sorts of scales. I don't know how this fits in with what the others were describing. I don't know whether the other fields also have this scale of things that goes from really, really tiny things to really humongous things that are built out of these really sort of incredibly small pieces. The things that fit together, it's making all the pieces fit together, which is tricky for us. I don't know whether that is something that other people sort of feel is something they also face.
I would open this up to the audience, by the way, because what you described is a lot of similar problems to what people are working on with ontologies. Does somebody who's really a hardcore ontologist, is Sebastian still on the line? I think he'd have a great answer to this.
So Gene Ontology has a meeting. So I think people who are starting with ontology, it is a great meeting. And then Plantium project for the plant people, we also host from time to time. And I think we could perhaps, survey what needs are there. And it may be a good idea to have a dedicated kind of a YouTube channel. We already have for ISP, but people can make little short videos, 5 minutes, what ontologies are, how you use it, how to find it, something like that.
I will just throw in there that there is Obo Academy, which has a lot of courses on working with, and just to get back to the basic question, ontologies in the sort of simplest definition, it is a structured vocabulary where all the terms are defined and they have defined relations between them, and you can reason over them. So you can go from very, very granular, like the leaflet of a heart valve, all the way up to the very large of like the whole cardiovascular system. So it is sort of taking it, and people can come in at whatever level they want and know that if I start at the heart morphology, I'm going to get everything below that. I'm going to get all, if it's Uberon, I'm going to get all the parts that make up the heart. So it is similar, but the key parts are everything should have a definition so that you don't end up arguing forever. What do you mean when you say gene or allele or heart? And everything has to have a defined relation where it is always correct to reason up. So the lower thing, if you start at heart, everything below that is part of the heart.
Okay, yeah, so this sounds similar to, in math, there are definitions, of course, and then you sort of prove facts about those definitions, and you sort of realize that what you thought had some properties also had other properties. And in fact, one of the advantages of digitalizing mathematics is that you can sort of really go back to the very sort of far beginning of things. And of course, in sort of informal math, people have slightly different conventions in certain areas, whereas when you formalize things, you sort of agree that there is no alternative, it's just this, and then you can see what choices have been made. So this definitely is something that resonates with what we're doing.
Right. I see there's quite a bit of discussion in the chat. I think I saw earlier somebody had raised their hand to ask a question. Is that still of interest? Otherwise, I have some more questions. Let's see. Well, something that's very interesting, this is a little bit of a different direction. What is the funding situation like for the curation efforts in your field? So I think this goes along with what we discussed earlier and how people become curators or how they get into this. But, what parts of those efforts are funded in your field versus what parts are in-kind contributions that are part of bigger projects?
Maybe I can start. So
Please.
until a couple of years ago, it was mostly only voluntary. But in the last couple of years, big AI companies have become interested in this. So there's been a lot of funds thrown into the field. And so a lot of the funding right now is from either big AI companies or big philanthropic associations that donate large sums of money with specific projects in mind. Some of them in the form of grants or sort of grant-like things, others in the form of hiring contractors to sort of perform some part-time task. And other companies are just hiring people that are experienced in this to develop their own specialized AI agents to sort of solve mathematical problems. But this is something that has been very recent and within the last 2-3 years at most.
So maybe a follow-up question on this, because I think the idea of doing theorem solving with LLMs has maybe come into the popular conscience. And on the biology and the biocuration side, there are all sorts of really high-profile drug discovery ideas where people are using LLMs now. So DeepMind has its AlphaFold thing, which I would say is one of the most high-profile uses of resources that have been curated. There's been a little bit of a disconnect between these resources and the technologies and the infrastructure that built them and the people who have curated the data that went in. I would say that across our community, we probably haven't reaped the benefits of funding from the large companies that have been using the results of these. And there's a very big discussion about this. But Damiano, how do you think that biocurators could tap into these kinds of resources, which seem to be quite interested in what you're doing right now?
So for us, I think, at least for me, it still looks very fortuitous. I don't really know why people became interested so much in sort of formalized mathematics. I would like to think that it is because large language models make lots of mistakes all the time. And at least with computer programs like Lean that are sort of precisely designed to verify reasoning, you could get an LLM to ideally sort of say something and then verify that what they said is correct, at least in the scope of things that can be verified correct. And mathematics is a sort of perfect playground for this because in math you can really be right or wrong. So you either have a proof of something or you have a proof of a sort of negation of the something. Or I mean, you could have neither, but in theory you might be able to have exactly one of the two. And so if your LLM makes a claim and then it's able to prove it by, let's say, a mathematical claim, and then is able to prove it using Lean, then it can just say, well, this is true, and you don't need to argue with it. So I think this is the main reason why AI companies became interested in this. I don't really know what they gain by doing this, which is maybe a little bit worrying because I don't know how long this will last and whether it's just a sort of fashion right now and it's going to last a few more years and then it's going to disappear. But overall, it seems that there's a lot of interest into seeing how much you can push artificial intelligence and large language models to really do stuff that people have been doing by hand for a long time. So I would imagine that something like this would also be of interest to companies, and probably the more sort of human-like the endeavor, the more they would be interested in seeing that computers can also do this thing that really looked like a human prerogative. Maybe this is what attracted people to mathematics, because people were saying, oh, reasoning is really something that machines cannot do. And then, well, they still cannot do it, but now they're at least now they're saying wrong things instead of not saying anything at all. So I guess that's an improvement.
Great, thank you. So the takeaway is we need to look, every community, not just biocurators, need to look for the tasks which can be evaluated easily and have this sort of like tantalizing applications. So
I think.
Sorry, go ahead.
Yeah, so I was going to say, I think having clear measures of this is correct, this is incorrect, it's very good for computers, because then they can train. They can make an attempt, they can see that whether the answer is right or wrong, and if it's right, they continue doing their thing, and if it's wrong, they do something else. So I think having clear goals is probably very important for machine learning.
All right. I think that's a good place to leave it for our panel discussion. Thank you very much to our panelists for giving your insight from your different fields. It's a lot for us to think about. I'm going to pass it back to Federica, and then we have one more session, part of the session before we close.
Thank you so much, Charlie. We now have our long talk. Speaker is Emilie Pasche. She's going to talk about from SIBiLS to BiodiversityPMC, a PMC superset with advanced functionalities. So Emilie is a research associate at the Swiss Institute of Bioinformatics and the University of Applied Sciences of Western Switzerland. And she's a part of the team behind SIBiLS, a semantic literature service supporting customizable search and curation workflows in the life sciences. And she also contributes to the development of the user interfaces for the SIBiLS tools. So thank you, Emilie.
Thank you. So can you see my screen?
We do, yes.
So I'm going to start with a quick introduction on SIBiLS, and then I will present you BiodiversityPMC. So SIBiLS is a short name for the SIB Literature Services. So it's services to collect publication from multiple sources, such as Medline, PubMed Central, supplementary data, and so on. And we are annotating all these data, structuring them, and finally using them to provide services that support research. So we have several services like BiodiversityPMC, Variome, etc. So I'll briefly show you the architecture of SIBiLS. So we have a process that is doing it in backend. So every day we are collecting documents, new documents. So it's citation, full text article, PDF, images, tables, taxonomic treatments, and so on. So we take very heterogeneous formats, and we are parsing them to more uniform content. Then this content is mapped to different terminologies. So actually, for the moment, we have more than 20 vocabularies, such as the mesh, the gene ontology, UniProt, etc. And we are using all the concepts to map the terms, the synonyms to our content. And then everything is pushed in a database, a MongoDB database. And on top of this database, we are building Elasticsearch index, which is used for our services. Then we have the front-end part, which is composed of web portals that are our services, so BiodiversityPMC and so on. And we also provide access to the API. So for instance, the search and the fetch APIs. So if we have a look at the collections, we started SIBiLS with two collections, which were Medline and PMC. So Medline, it's the abstract mainly in life and health science. And actually, now we have 40 million abstracts. And PMC, the full text articles, and we have 8 million abstracts. Then in 2021, we have added the supplementary data. So the supplementary data, it's data that mostly are not included in the main body of the article. So it's images, it's Word documents, tables, PDF, and all these contents, we are processing it to extract the textual content so that we can search in it. And now we have 30 million supplementary data. Then in 2022, we started including PLATSI, so it's a taxonomic treatment, so more biodiversity data, and we have now 1 million. 2 years later, we have updated PMC on our site, so it's no more only PMC, but it's what we call PMC Plus. So it's a full text article not only from the NLM, but also from different journals such as Pensoft and EGT. And we have now like 30,000 documents from these new journals. Then last year we have started integrating some Zenodo communities. So it's communities that are from the biodiversity fields. And it's very different, very resource type, like we have PhD thesis, conference paper, data set, and so on. And the future of SIBiLS, we are starting to try to integrate OpenAlex content. So it's work in progress. We don't have yet a date for when it will be available. Then we do the annotation. So the annotation, it's for quite a known reason. So, you know, in the literature, there are many terms that are used for the same concept, synonyms, abbreviation, old or variant names. So with the annotation, we are creating consistency. And like this, when you do your query, you will find also documents if synonyms is used. So we are using various terminologies. So here you have the list of the terminologies that we have for the moment, grouped by a type of research. So we have terminologies for the molecular and cellular biology, such as cell ontology, Cellosaurus. We have terminologies for gene and protein, for health, for fair data, and we have quite a lot of terminologies for the biodiversity fields. And we are updating, adding new terminologies on a regular basis. So SIBiLS, it's a kind of umbrella for several text mining services. So today I'm going to present you the biodiversity PMC services. We also have question answering services where you can ask question and get a short answer together with the passage where the answer comes from. We have also triage services, which is kind of a weekly digest of the literature. So every week we are processing the new documents that have been added to SIBiLS, and we rank them according to certain tasks. So for instance, we have for the island biodiversity or for the Cellosaurus. But the triage will be presented by one of my colleagues, Paul van Heijden, in a further virtual session. I think it's in September. We also have Varium, which is for searching for genetic variants in the literature, and Biotics Explorer for the biotic interaction in the literature. So all these services are built on top of SIBiLS. So today I'm focusing on Biodiversity PMC. So Biodiversity PMC is a search platform which gives access to a wide range of content in life and health science, as well as biodiversity and related science. So it's targeted for all the scientists, such as life and health science, ecologists, environmental researchers, etc. So I will give you a quick overview of the tool. So here you have the URL if you would like to test it, or you can scan the QR code. So we have deployed it today, so it might have a few bugs. If it's the case, please contact us so that we can check what happens. So when you arrive on the front end, on the landing page, you have here the list of our services, so you can jump to another services if one is more useful for you. And then you have the research bar here, and on the bottom of, in the middle of the page, you have some statistics about our collection. So it's daily updated, like this. You can see where we stand with the collection. For some of the collection, we also provide numbers for the sub-collection. So for instance, for PMC Plus, the PMC OA subset is the NLM PMC. You have also the author manuscripts provided by the NLM. And the PMC Plus subset is the articles from the biodiversity journals. So if you want to use the service, you can type your query in the search bar. You can also click on Show Example to see a few examples of type of query that you can do. And you also have some links of queries that will be redirected to other of our services, for instance, Varium, or the question answering. So let's try with the P53 transcription factor target genes query. So you click on the search, and you will get the results by collection. So you can see the result in Medline, in PubMed Central, in supplementary data, and so on. And you can also use filters to narrow down your research. So you have the filter on the left of the screen. And we have three types of filters. We have temporal filters, so you can search for a specific date range, or for document that has been released in the last days or the last months, and so on. You can also search on the general filter. So for instance, if you are interested in a specific article type or a specific language, license, and so on. And then we have the annotation filter. So we have all the terminologies that I showed before, and you can select the one that are of interest for you and a filter for one specific concept. So by default, most of the filters are hidden, and you can select the one that you would like to use. Then you can visualize a document. So here, for instance, if you click on the link icon, you will see all links to our internal services and also to external services such as Medline for the NLM articles, or the publisher when it's a full text article. And you can also click on the title of an article to see it in full screen. The query terms are highlighted, but you can also highlight the annotation that we have in the document. So if you click on the small show annotation panel on the top, it will open an annotation panel where you see all the terminologies that have been annotated in the document. So you can select the one that are of interest. So for instance, here, all the gene ontology terminologies, and it will highlight you all the terms from the gene ontology. You can also export documents. So for instance, if the second document was relevant for your query, you can flag it. And on the bottom of the page, it will add it to the export box, and then you can export it as JSON or CVS for further use. So that's it for the interface. Now I will give you a quick overview of how to make a more advanced query. So we have two modes for the query. You can either use the manual query syntax, so you type your query directly, or you can use the advanced query builder. So the advanced query builder, it's to build a query using a more visual way. So you can add blocks, move blocks by drag and drop, and so on. I don't have much time today, so I will not show you the advanced query builder, but I will focus on the query syntax. But you can do the same with both interface. So regarding the query syntax, by default, when you type a word, we automatically expand it. So for instance, if you are searching for prenylation, we will not search only prenylation, but also the corresponding gene ontology term and the corresponding mesh term. So you will get all results that contain either the word or one of these concepts. Also, if you are typing some stop words such as the, of, and so on, they will be automatically removed from your query. If you would like to have an exact sequence of words, so you don't want that we remove some words or that we expand your query, you can use the double quotes. So everything which is between double quotes will be searched exactly as you type it. So for instance, if you type protein prenylation, you will get the paper on the left of the screen, but not the one on the right, because both words are not together. You can also query for semantic annotation. So you can type the name of the terminology, double dot, and the identifier in the terminology. And it will search for all documents that have this annotation. So in the manual way, it means that you need to know the concept identifier. But if you are using the advanced query builder, you have some user-friendly way to find the term without having to know the concept number. You can, of course, use all the Boolean operators, so and or not, and as well as the parentheses to build the more advanced query logic. For instance, you can search for P53 and apoptosis, and it will search postword in the document. You can search in a specific field, so you type the name of the field, double dot, and what you are searching. The list of the fields that are available, you can find them in the documentation page. Or if you are using the advanced query builder, you have the list of the fields in a drop-down menu. You can also do fuzzy search if the terms that you are searching can be spelled slightly different, or if there are frequent misspelling on system. So then you can just use the tilde after a term, and it will edit the distance. You can also specify the distance that you would like to search. All this is described in the documentation. You can also do proximity search, so it means that you would like two words to appear close to each other, not necessarily next to each other, but close. So by this, you use the tilde and the distance that you allowed. So for instance, if you put distance 2, it means that it can have two words between each other. You can also do range search, so it's specifically for the dates. If you would like to search, for instance, articles that have been published between one date and another one. And if you can use the wildcard if you want an open range. So that's it for the query syntax. Everything is also accessible through APIs. So we have some of the APIs are the search, where you can do a query and retrieve relevant document for your query. We have also the fetch, where you can retrieve a document together with its annotation. The QA for natural language question. The query player that enable you to build an Elasticsearch query so that you can then search it in the search API. And all these API will be available and described on our website. So as we have done the migration to the production today, it's not yet updated. So the description of the API online is still on the old version. So it will be updated very soon.
Quick note, oh,
sorry?
Go ahead.
So to finish, I would like to thank all the people from my group who contributed to that, our collaborators, as well as the funding. Thank you very much.
Thank you so much, Emilie. It was really, really interesting. And I see that we have a couple of questions in the chat. And I wanted to ask you and Tiago if you want to speak directly and interact with Emilie.
Yeah, I can ask. I have to leave very soon, though. I was just wondering when I caught my attention when you said you were pulling in the supplemental data. And I was wondering if you were doing that as an archive or as like a drop and reload. Because I found in the past where we've gone to look for supplemental data that the article says exists, but the journal has thrown away. So it's gone.
So I'm not totally sure about that because I'm not in charge of the collecting, but we are collecting usually a basis every year. So it means if something disappears, we probably don't have it anymore, neither. But then after we are updating every day to get the new one. So I need to check with my colleague if we keep what has been available.
Well, yeah. Yeah, just a heads up. We archive it for any journal article that we use, because otherwise it's just gone. And with so much of the methods now being in the supplements, it's a real problem if it disappears. Thank you. Thanks for everybody for putting this together. It's been a great session. Thank you, Charlie and Federica.
Thank you so much, Sue. I see that Tiago cannot speak right now, so here is this question. What is the relation of Europe PMC and the Biodiversity PMC? There seems to be a lot of synergies between the two.
Yes, I think, again, I'm not the expert for the question, but there are links for the annotation. I know that at one point, some of the annotation we were doing were pushed to Europe PMC. We have also links from our services to Europe PMC. But yeah, I'm sorry, I don't know exactly.
Okay. Yeah, I have a question. Sorry, I'm not able to raise hand. I don't know if you're finished with the previous. Yeah, it's fine. Okay, the biodiversity system you are building does also consider biodiversity data, because the example was more biomedical data. So some zoologists would be very interested in this if it really consider, I don't know, ecological data or.
Yes, it is biodiversity data. We have both. We have the life science from the NLM, and now we have the PLADSI treatments, which are only biodiversity. We have the journals, for instance, PenSoft and so on. So we have a lot of biodiversity data. Zenodo also, it's only biodiversity. So yes, it's clearly for the
Thank you very
biodiversity
much.
field. Yeah.
Thank you very much.
All right. That's it. Thank you, everybody, for joining. Last notes before we go, and we're on time. We've got 3 things from announcements. Probably there's more, so please keep up with ISB on our social media, on LinkedIn. Please join our Slack if you're not already in it. A couple of things that have happened recently. We made a podcast where we're interviewing people about the way they do curation. We got our 2nd episode onto YouTube about the Disprot resource. If you are not already planning on going, consider attending the in-person conference in Cape Town. There's also a hybrid option. And the next session of our virtual conference will be the poster session in June 8th. There is a link on our website, and we will share it with all of you on how you can make a submission to be part of the virtual poster session. So with that, I would like to say thank you to our speakers, thank you to our panelists, thank you to Federica for being my co-organizer, and we hope to see you all very soon. Bye-bye.
Bye.